All right. So I wanted to start off with kind of going over the survey questions that were from Homework 1. So thank you all for giving answers. I did read through every single one of them, and I tried to just kind of summarize and put some of the big ideas together, and I will talk to you about them today. So why are you taking 315? For a lot of people, it was a major requirement or a prerequisite for whatever program that you're applying to next, whether it's graduate school, med school, pharmacy, physician's assistant, dentistry, whatever it was, right? There are a few people that said that they were taking it as a biology elective or they're retaking the course. Some people just out of general interest in microbiology, or because maybe you're in a research lab on campus and in one of the micro labs and it's going to help you out with your research. And then a few people also were interested into just general interest in, again, in learning more about microbes, and a lot of people were interested in viruses in particular. So what do you hope to gain from 315? Again, a lot of people had this similar kind of answer about just general knowledge about microbes and how they function, but kind of some of the different contexts were wanting to know how the roles that they play in the environment, and then in like with human health, and then also just wanting to know like real life applications of this, and then how can we actually use bacteria and viruses to help humankind? And then a lot of people were just, again, general knowledge in preparation, you know, whether it be this is a prerequisite for a class that you need to take, or if you're going to be able to use this for your career. All right, so one of the big ones, what concerns or apprehensions do you have about taking 315? I think some of the biggest ones were a lot of you have heard that this course is very hard and that there's a lot of material. So basically like time management and quantity of information were big things as far as worries about this class. So I guess what I will say about that is, I said this in the first class that yes, we are going to go through a lot of material. I think that in itself is what makes the class difficult. I don't think it's difficult for you to be able to understand each and every individual topic that we're going over. And if you are having trouble, please ask questions, stop me. I will repeat things over and over again, try to get them in different ways until you understand it. Come see me. But yes, I am going to be honest, we do cover a lot of material in this class. And so a lot of it is just going to be keeping up and trying to figure out how to manage doing that. And so again, you know, it's going to be different for everybody how you do that. And so if you want to come talk to me about it, I can talk to you, offer some suggestions and things like that. But my biggest suggestion up front is to just keep on top of things, and that's kind of why I have the readings before class with the homeworks before, and so that you are at least getting some of it in before you come to class, and that kind of helps so that you're not just trying to learn it all right before the test. Some other worries were memorization and essentially lots of, you know, the bio terms, the bio jargon, and the definitions. So again, I'm going to be honest, this is an introductory level course, and so there are a lot of terms that are just going to be memorization. Again, there are different techniques that you could try, especially for, I would say, the first and the last section of the class. There's going to be a lot of, you know, just memorize this scientist and what did they do. And then at the end, when we're learning about the different applications and things, there again is just learning a lot of different things. In the middle, I hope that there are more chances for understanding, and we will try to get through different processes and things like that and trying to put big pictures together. So the two mid sections will be more understanding based. And then as far as for tests and how to study, I will, before the test, like kind of send out a note as far as what the question types are going to be, so you know what kind of questions, at least the format of the questions that are coming at you. And My biggest tip is to use the intended learning outcomes as your study guides. And so essentially that is where I'm going to look for when I come up with the questions. Again, we mentioned this before. Pace of the course, yeah, because we do go over a lot of material, we try to go through it quickly, but again, please stop me. If we don't get to something, I will decide at the end of the next day whether I cut it completely or we just go over it to the next class. So don't worry if we keep talking about something, that's totally fine with me. Lecture capture not working. So like on Monday, the mic wasn't working, so you wouldn't get the official lecture capture, but I don't know if you've seen, like I said, I am recording what I'm doing on my iPad, and that has been posted, and so hopefully both won't fail on you. Relating different processes, hopefully I'll be able to help you out with that when we do get to like we're talking about the different systems within the cells. For some of you, this is your first bio class in a while, or maybe the first bio class at CSUN, so a little bit worried about getting used to that. And then for some of you, you didn't really have any worries, so that's good. All right, so what are some of your expectations from me? The biggest were essentially that I convey topics clearly, so explaining things well. I will ask for your help in this. If you don't understand, I won't know unless you point that out to me, so please do. Being open to questions, hopefully you will see that I am very open to questions and also very open to coming to office, having you come to office hours, making appointments. I will try my best to make connections to real life applications to help you figure out like how to, just not this random concept that we're learning. Making lectures engaging, I'm still working on that, so we will try. The first time through, but the material was a little bit tough for me, but now that I've done it once, I'm hopefully will make that a little bit better for you. Slides prior to class, I always try to do it at least the day before or the night before. If come, Monday morning, Wednesday morning, you see I don't have it, please email me, because there is a chance that I just thought that I did and I didn't. That happened once last semester. The study tips and study guides. So again, for the study guides, that's essentially the intended learning outcomes. Study tips, the biggest thing I said, again, for starting off, is for you to just keep up with the material as we go, because there is a lot of it. But as far as things work differently for everybody, but I'm happy to just, kind of shoot back and forth ideas with you as far as how you study for your other classes, and is that something that's going to work for this. Fair grading, I always try to be fair in the grading, and in a lot of cases you'll see half of the tests are multiple choice, so I try to come up with fair questions, and if it does turn out that there was a question that was worded improperly, I have taken questions off the exams for that, or given essentially everybody points for that question. Example problems, so the example problems, most of them were going to come from the homework, and then essentially these Kahoot questions, which I will go ahead and actually post the questions for you on Canvas as well. And then another thing was an organized Canvas page. Hopefully it's organized enough for you. If you have other suggestions, please let me know. I essentially do everything by type, so all the homeworks are going to be under one thing, all the the actual lecture slides are under one, the recordings are under another, and so hopefully that's easy enough for you to find. But if it's not, please, I am open to suggestions. So I went ahead and essentially answered my version of the questions that you had, and so I'll share that with you. So why am I teaching 315? So like I said, I'm still new-ish. This is my second year here. And within the micro faculty, all new faculty essentially we start teaching with this class, because this is like our core class for the micro department. And we usually like to be able to teach something at least, more than once, so that we can improve and get to know that class better. So that's why I'm teaching this. What do I hope students gain from 315? Essentially a solid foundation of microbiology principles, right? Again, we're going to learn a lot of different things, but hopefully, you know, it's just the basis for the things that you're going to need later, whether it's in your careers or in another class. And so at least hopefully if you've seen it once and kind of understood a little, even if you forget, you know, over winter break or over whatever, the next time you actually need it, it should be easier for you to come back and remember and learn it again the next time if I can give you that good foundation to start. The other thing is that I hope you learn, or at least think about how you learn. And like I said, I'm also big on helping people figure out what's the best study techniques for you. So please come talk to me about that. What are my concerns or apprehensions about teaching 315? So I am a little bit worried about being able to explain something in a way that every single one of you understands. So again, please let me know if what I'm saying doesn't get through to you. And even if I can't get it through in lecture, I will take the time to go back and think about it, and maybe I can talk to you after class and come up with another way to make things clear. I guess general other apprehensions, you know, I'm kind of a introverted, shy-ish person, and speaking in front of large groups still makes me very nervous every day, so still getting used to that. What expectations do I have of the students? Essentially, come ready to learn and be open-minded about the topics that we're going to learn, and then just general patience. You know, these rooms are very technology-based, and hopefully things will go smoothly and things like that. But hopefully I can get help from you with that. The other thing that was super nice about reading the surveys, that it sounded like a lot of you are really excited to get started in this class. So that's good. That's good for me to hear. That, and hopefully we will keep that excitement up going through the rest of the class. All right, so we'll get started with the actual lecture material for today. So again, trying to get you used to these intended learning outcomes. So this is what we're going to go through today, is going through spontaneous generation and how it was disproved. We'll go through Koch's postulates. And then again, so for the rest of this, and so the chapter 1 is a lot of kind of going through history and how microbiology was founded. So we'll go through some of the more famous scientists that have contributed to microbiology. And again, as a reminder, there are, especially in today's, there's going to be more scientists than I expect you to memorize for the exam. So only the ones in bold are what will be on the exam. All right. So the first question, so we had left off essentially with Hooke and Van Leeuwenhoek is having the ability now to look at and see these microorganisms, right? And then, so the question then was, where do they actually come from? It turned out that the chemists at the time had thought that they actually came about by this term called spontaneous generation. Essentially, that they just kind of appeared out of nowhere, right? That there were just these tiny things, and they could just poof, and there you go, and that's where they would be. It turned out that, you know, that a lot of actual, so Reddy and the next person that we'll talk about were actually priests. Then got involved because they didn't like this idea that you know, there's something that just could come out of nowhere, right? They believed that things actually came from parents, that there had to be a parent, and so they really got involved in kind of these scientific things. And so the first person that we'll talk about, Reddy, he was actually, and a lot of, or in the beginning, what they were studying were noticing, one of the first microbes that they noticed were maggots. So these are essentially fly larvae. So he was studying the appearance of maggots on decaying meat. So he set up this experiment and had 3 different cases with meat in different jars. So the first case, he placed meat in an unsealed jar that was open to air. Then he placed meat in a sealed jar, so like with a cap on it, so nothing could get in. And then the third case, he placed meat in a jar, but he covered it with gauze. So it's like large physical things couldn't get into it, but air could still get into it, right? So then what essentially we're looking for to see, you left it out, and then you wanted to see, did the maggots appear in the next couple of days or whatever. I don't know the exact time. So think about it. What do you predict would happen in each of the different cases? Do you think that there would be maggots in the case of the unsealed jar?
Yeah.
Okay. How about with the one with the lid, like a screwed on lid that was tight?
No.
Okay. And then what about the one where there's just kind of a breathable gauze over the top of the lid? Okay. So, that's a good question. I'm not actually sure. Small enough so that actual flies couldn't get in. But okay, so this is the picture of what actually happened, right? So again, so the one in the open jar, that's an easy one, right? The flies could get in, and so they would lay their eggs, and so then you would get the maggots on them. The case where with the sealed jar, nothing could get in, and no flies could get in, and so you didn't see any maggots. And then in the final case, again, you actually didn't see any maggots because the flies couldn't get in. They kind of pictured that, there were maybe the maggots on the top. So the basic conclusion that Reddy was able to make in this context, right, that the maggots actually came from the flies, right? It had to have been the flies in order to get the maggots. And so therefore, maggots did not appear by spontaneous generation. Okay, so that was what he proved, but now essentially what happened in every case, so even in these two cases where there were no actual maggots, the meat still putrefied and you would get bacteria, right? So then the question became, okay, so spontaneous generation does not apply to maggots, but how about microbes, like other bacteria? Okay, so the next person that came into the picture again was another priest, so Spallanzani. So he did a different experiment. So by this time, they had realized that if you boiled media or the broth that they were testing, that would kill off things, right? That was a form of sterilization. They had figured that out at that time. So what he did was essentially he set up two cases. One where, again, always starting with the sterile broth. So they would boil the broth in each case, and then he put one in a flask that was open to air, and then one in a flask that was sealed, so no air could get in. And so the one that's open, obviously, he got a bunch of microorganisms growing in the broth, and you could tell that by it was cloudy. You could see it in there. And then, but in the case where the flask was completely sealed, he couldn't see any growth whatsoever. Okay, so this, again, has his thought about disproving the idea of spontaneous generation, right? Because there was nothing in here, nothing appeared. But then the question became, so what about air, right? So the question was, what if, in this case, there's absolutely no air that was allowed into the flask. What if air was actually required for them to grow? So even if it were to come out of nowhere, it couldn't do it because there was no air. And so it wasn't actually until Pasteur that we were able to finally disprove the idea of spontaneous generation. So he invented this special swan neck flask. So it has this shape here. So you would have the media, again, you would boil it before to sterilize it, put it in this flask, and essentially this S shape, this swan neck shape here, would allow, so you'd have a tiny opening on this end, and that would allow air to come through. So the air can come through, and it will come through here. And so that would make sure that if air is required for these microbes to grow, that they had it. But what this actual S-shaped portion of this did was it would actually trap all the dust and anything that was actually just flowing through the air from getting into the medium here. And so you could let this grow for a long, long time, and you wouldn't actually get any growth in here. So that was the kind of final disproof of the idea that spontaneous generation of these microbes was a thing. Right? Okay, but as always, there are always exceptions. So after Pasteur's experiments, you had all these people trying to go and repeat the experiments, like good scientists, and try and figure out, and did that actually work for them? So in the case of Tyndall, what happened with his experiments was that he actually observed growth regardless. So he was working and he would boil the broth that he was trying to do, and regardless of how long that he boiled that thing for, he would still get growth, right? But he found that if he did cycles of repeated boiling and resting, so he would boil it and then let it sit for a day, and then boil it again the next day, and then let it sit, and then boil it again, that he would actually be able to get to a state where there was no microbial growth. It turned out that Tyndall had discovered were these endospores, which we will talk about kind of more towards the end of the class, but you've probably heard of things like anthrax, right? So endospores are essentially these dormant forms of cells that are resistant to heat. So that is essentially why he couldn't get it in this case. But any ideas of why this kind of boiling, letting it go, boiling, letting it go, why did that actually kill the microbes? Yeah?
Because once the first boiling, they don't die because they're heat resistant, but then when it cools down, they come out of their shells and begin to be active. And once they're out of their shells and active, the heat kills them.
Yep, exactly. So in case you didn't hear, so what happens, so these endospores, again, they're dormant forms of cells, and they go into this form when essentially there are like no nutrients enough around for where their original cells were to grow. And so they go into this form, they become very resilient, and so now the media gets boiled, essentially everything else is gone, right? And so now there's tons of these nutrients for them to eat, and it comes and it essentially wakes up, and then it kind of goes back and it germinates into the normal form of the bacteria. But now Tyndall went ahead and then caught them in its normal form, and then boiled them again and caught those guys and killed them off. All right, is that clear? Yeah.
So it has more to do with the availability of nutrition than it does the change in temperature of cooling after the first?
Yeah, essentially that was allowing the cells survive, the spores would survive that is heating, right? Once it cooled down, that does play a role in, like, because that's when the normal growth temperature of the actual active bacteria are, so that does play a role, but it comes back out to its normal form because there are these nutrients available. All right, so that was kind of the story with spontaneous generation. Now we'll move on in history, and now people are trying to figure out, well, now we can see all these microbes, we know they exist. Now they began to become suspicious that they actually cause disease. And it didn't necessarily have to cause disease in humans for them to get concerned, right? So the first kind of cases where scientists figured out that there were microbes causing disease were actually in terms of fungus, whether for silkworms and for potato blight. So it wasn't actually until Robert Koch who actually came up with these, what we will learn as Koch's postulates, that he came up with a method to actually go through scientifically and figure out, is a certain microbe the cause of this specific disease? So he's actually known for studying anthrax and determining the chain of infection with anthrax, but most notably for determining what caused tuberculosis, which he won the Nobel Prize for. Yep.
Sorry, so did they prove that they actually cause disease or not?
Yep.
So usually, most of the times, microbes do cause.
So let's not say usually. There are many friendly microbes that don't cause disease. In this context, back when they were trying to figure out which one and what actually caused the disease, there were many cases where they were able to find that to be true. All right, so we'll go through Koch postulates. Again, so these are essentially steps that you have to go through to figure out, does this microbe actually cause the disease that you're observing, right? So the first postulate is that the microbe is found in all cases of the disease but is absent from healthy individuals. So that's a pretty easy one, right? So you're going to be able to find this microbe in all the sick people but not in the healthy one. The second one, the microbe is isolated from the diseased host and grown in pure culture. Okay, so this one required a little bit more. So for you to be able to, with absolute certainty, say that it was this one microbe that is causing this disease, there has to be a way for you to take it out and culture that one microbe by itself, right? And so this is what is known as these pure culture techniques, and we'll learn about these more in lab, just culturing in general, in the next couple of weeks. But so these actual pure culture techniques that were kind of coming along at the same time as Koch with other scientists, is very key to be able for him to set up these different postulates. So when the microbe is introduced into a healthy, susceptible host or animal model, the host shows the same disease. So the idea is that, okay, you have the sick population, the healthy population. In two, you were able to take out that microbe from the sick population. Now what has to happen is you're going to go and give that one to the healthy population, and that healthy population has to, again, become sick from the thing that you isolated. Okay? And this kind of leads into 4. So the same strain of microbe is obtained from the newly diseased host. So when cultured, the strain shows the same characteristics as before. So now you've taken it out, put it into a healthy host. That host becomes sick with the exact same thing, and you're able to then take that out from this newly infected host, and that microbe has to be the same as the one that you got in number 2. Okay? So then we're going to keep talking about this. The next slide is just kind of a pictorial view of how this happens. So in this case, studying something in rabbits. And so you have a healthy population and a sick population, and you are able to take out the microbe from the sick population, but nothing from the healthy population. Again, so this key is that you're able to then go and take these microbes and grow them in pure culture. So that pure culture meaning that it's just that one microbe, so that it can't be mixed with more than one in there, because they wanted to know that this was the one thing that was causing the disease. All right, and then so now you take a healthy rabbit and you give it these microbes, and it should show the same symptoms as before, as in the first case. So this rabbit now becomes sick. But then again, kind of wrapping it up, you're also now from the sick rabbit able to pull out the exact same microbe that you had before. All right, so this was great. Back then there were a lot of, and we'll see a list on the next slide, that there were a lot of different diseases that they were able to pin down to different microbes for this. But just as an exercise, can you think of a case where problems could occur with trying to implement some of these postulates? You can think about it, talk to each other if you want, and then I will take some answers. Or if you're ready, if somebody has an idea. Yeah?
You don't have a way to grow you don't have a way to culture the microbe yet, like you don't know what intermediate to use and what conditions to use.
Yep, that is a very big problem, right? So number 2 is a very big problem. If you don't know how to grow it, we can't grow it. There's no way to absolutely pin that down. Yeah.
If it's like a microbe from a human, for number 3, like it's unethical to put it, inoculate it, and then get it up in the human again?
Yep. So you essentially need a model, right? An animal model for you to be able to put this in, right? So, and we're not, okay, so back in the 1700s, the scientists were happy to kind of test them on themselves. It's crazy. There were a lot of cases where they would actually test things out on themselves. But that's kind of unethical. We're not going to be trying to give humans these things, these diseases. So in some cases like that, right, now we have kind of tissue culture techniques now. And so it's not the full model, but people are kind of relying on being able to reproduce that kind of infection on just the cellular basis. Anything else? Yeah.
Sorry, so I'm confused. These bullet points are actually the problems?
These are the postulates. These are Koch's postulates. This is like the method that the scientists use to go through and step through to be able to identify the causative agent. Yeah?
When you're trying to infect a healthy host, it has built-in immunity of some sort already?
Yep, that could definitely be the case, right? Because then, so that's kind of in this step 3, right? You're kind of trying to give it to the healthy host, and then they're immune and they don't actually show those symptoms again. So that's going to cause a problem. Yep.
Number 1, I mean, when bacteria is in their dormant state, you won't be able to. The people will seem to be healthy, but they actually have like bacteria living in them.
Yep, exactly. So what we're kind of talking about now is that there are some cases where in step 1, right, it would have to have been the case where you have the healthy individuals or animals, right, don't have the microbe that you're talking about, but the diseased ones do. There are cases that we know of now where you can just be carriers of bacteria and they're just chilling and they're not causing you any kind of problems, and so it's going to show up in you. And the one example that I know of the best is in C. diff infections, right? So you can be a carrier of C. diff and it's not going to give you a problem until the conditions are right. Yes, sorry.
I think one of the counter arguments to the possible answers for number 4, it's like where it says the strain shows the same characteristics. There's always the possibility of a mutation that can make it stronger or weaken in certain areas.
Yep.
If it's like non-synonymous and it actually happens.
Yep, exactly. That is a very good point, and that can very well happen, right? So you put it into a new host, it's adapted to its new host, it's come up with new characteristics, and now it's not the same. But it was the thing that actually caused the disease. But now, if you're trying to really, really be tight on this, that's not going to qualify. You had something else?
Oh, yeah.
No. Yep. Yep.
So when you said that you might have the microbe, but it's dormant, does that mean that you don't have the right receptor for it to be active in your body?
It depends on what that microbe's doing, right? So in the case of C. diff, for that to make you sick, it's going to be producing a toxin. And so it's only going to produce that toxin when the conditions are right. And some of them actually don't even produce the toxin. And so it just kind of depends on what
For number 3, it says the host shows the same disease, but it could interact with people differently.
Exactly. Yep. So not everybody is going to be the same. You might end up with something different.
Did we
Yep.
mention for number 2 did we mention that not everything can be grown in pure culture?
Yeah. Yep. So number 2, the key for this is knowing how to grow things in pure culture. You have something?
Back then, did they use animals or did they use people too?
Mostly animals. I guess way back in the day, like I said, people volunteered and tested that, usually on themselves, not on others, but usually animals. Did you have something? Do you have something? No.
If the microbe is a virus, it won't grow in pure culture?
Yep. So there are separate techniques that we know of now to be able to culture viruses, but again, back then that they wouldn't have been able to figure that out. Did you have something? No. Okay. Was there somebody else over here? No. Okay. All right. Does this all kind of make sense? This is just kind of how they went through and kind of just like a scientific method for being able to do this. But as we know now, while this is a great kind of way to start, there are a lot of problems with this that we have to, with what we know now is going to be a problem.
Sorry, I had one more. When
Mhm.
when you're culturing, sometimes the colonies won't show because maybe the culture doesn't have nutrient sources for
Yep.
the whole community, so it just doesn't.
Yep. Again, so in the case of this is that pure culture technique is a really big one. Sometimes, sure, it might be that one microbe that's causing the problem, but it can only grow when it's surrounded by these other 5 different microbes, right? And so that would be very difficult to be able to discover in this method. Okay, good. Those are all very good, good ideas. All right, so like I said, so don't worry, you're not responsible for this, but I just wanted you to see that essentially, right around that time when Cook came up with all these postulates, that kind of just flooded the way for all of these people to go ahead and figure out what were causing all these different diseases. All right, so now we're going to move on to the first vaccines. The first story of vaccines kind of comes into play with smallpox. So in this 18th century, smallpox was a huge problem in Europe, but it was less of a problem in Asia. And so in Asia and Africa. So what was happening was essentially people were deliberately infecting their children with with smallpox you get these pustules. And so people decided that they were deliberately infecting their children with the stuff from these pustules, and that would essentially what we now call vaccinate them from and prevent them from getting smallpox. So what was happening is essentially the smallpox, it's a virus, was what we now call being attenuated. And so when you take them out of the pustule, they were essentially being attenuated, so they're like becoming weaker, right? And so that kind of weak form of the virus was then being put into the children, and that turned on their immune system and prevented them from actually getting the symptoms of the major disease. But back then wasn't perfect, and there were still cases where kids died from this. Yes.
Why was it more common in Europe than Asia?
It's not that it was more common, it's just that practice hadn't made it to, so I don't know, for whatever reason in like Asia and Africa, they started experimenting with giving these kids the smallpox was common all over the place. It was just that this method of using the stuff from the pustules and giving them to the kids. All right, so Lady Mary Montagu, so she was actually traveling through Turkey with her son and was experiencing this situation, the smallpox epidemic, and actually learned from the people there how to do this technique. And she had it done on her son, and she learned from that and then brought it back to Europe, and that's kind of how it spread in Europe. A different kind of approach to this was so in this case, they were actually taking pustules from smallpox itself. The other approach was with Edward Jenner. So they had found that the milkmaids that worked with these cows that were actually immune from getting smallpox, because they were kind of getting infected just by working with the cows. They were getting infected by cowpox, so the cow version of the disease. And so what they would then do in a similar fashion is, again, deliberately infect the patients with the cowpox, the stuff from the cowpox lesions, and that would essentially protect them from smallpox. And so this is actually where the term vaccination comes from, because it came from the word vacca, which is for cow. But there were people back then that were a little hesitant because they thought that if they would get something, the stuff from the cow, that they would turn into cows themselves. So, all right. So that was the first incidence of vaccines. But it wasn't until Pasteur, so Pasteur again, so he did a lot of things, that it really kind of started to take off, because he discovered what it meant to do attenuated strains, right? So making the microbes would be able to confer the immunity to disease, but then that they wouldn't actually cause disease in the different patients. And so he kind of started that whole kind of field and figured out how to get the attenuated strains for many different different diseases and different pathogens. One of the most important things that he learned was that, you know, every single pathogen was different. So he was actually trying to create the attenuated versions of these things. And so he had to go through a whole thing for rabies in particular. It required a very complex heat treatments and several different inoculations for that to actually work. All right, so the first antibiotics, the first antibiotic that we think of and known is penicillin. So that was discovered by Alexander Fleming. And this one is always the story of the happy accident in science. And a lot of times, you know, discoveries in science are made by accidents because you're trying to follow a protocol and you do something wrong, but then look, hey, this really weird thing happened. And that's how a lot of different discoveries are made. In the case of Fleming, he was studying a strain of Staphylococcus, but then he left his plate out. So this is what his plate, and this is all the Staphylococcus growing here. He left his plate out and it started growing this mold, and then he realized that this clearing zone here, so essentially this plate was full of staph, but now this mold started growing, and then this region out around it, that mold had killed all that staph. And so they eventually named that Penicillium notatum and figured out that there was a chemical substance that was coming out of that mold that was actually killing the bacteria. But it wasn't until 1941 that a couple of biochemists were actually able to purify penicillin from the mold, that we were actually able to widely distribute it and use it. And so it saved a lot of lives in World War II. So if you remember from Florence Nightingale, right, she had figured out that most of the soldiers were dying actually from infections from their wounds and not the actual wounds themselves. And so this was, in World War II, that was the first case where we were actually able to do something about it. And this kind of discovery led on to more and more discoveries as people just started looking at a whole bunch of different, you know, bacteria or whatever, and looking for the things that they made and see what might these things actually kill. And that's how a lot of the initial antibiotic discovery happened. All right, so the discovery of viruses. So again, you know, people were studying tobacco mosaic disease, right? So the tobacco plants, they were dying for whatever reason, and they were trying to figure out what was happening. They were kind of essentially, when you think about it, they were trying to go through Koch's postulates and figure out what the hell is causing this problem. But one of the ways that they were used to being able to collect the microbes was filtering them, right? So they would filter everything out and collect only the small things, or the things about the right size. But it turned out that whatever it was passing through their filters. And so this happened a couple of times, and they were actually able to conclude at that point that it wasn't a bacteria, because it was essentially too small, and it was just going through all the filters that they had known that caught bacteria. But it wasn't, so it took them a while, but then Wendell Stanley was finally able to purify the actual particle that was infecting these plants, and he got the Nobel Prize in Chemistry for that. And so he was actually able to do the crystal structure for it and figure out what the actual virus structure looked like. And we'll go into a bunch of virus structure later in the class. All right, I am going to stop here because I don't want to start this topic and move on. If